Now let's talk about the structure of the chromosomes and how they are condensely packed. Okay, so this is probably the chromosomal structure you can see through a microscope. Okay, so if you can maybe enlarge a segment of a chromosome, you may see something like that, a thread that are supercoiled, right, condensed structure. And in the length unit, maybe that's like a nanometer, 700 nanometer size. Okay, and then if you also enlarge that segment as well, then you will see these kind of structure. We call that about 300 nanometer structure. And then if you enlarge a segment, of course, you will see these. That's about 30 nanometer structure. And these kind of bead-looking things are so-called nucleosomes. Okay, so which has size about 10 nanometer structure. So nucleosome, a single nucleosome is made out of histone octamer. Octa is what, 8, and there's another one right here in between. And then so your DNA, the helical structure, goes around a single nucleosome twice. That's how a single nucleosome is made. So that's how you can package a long strand of DNA into very condensed form. Okay. So let's take a look. Let's take a look at the nucleosome structure. Of course, it's a building block of eukaryotic chromosome. Okay, it's got histone proteins. Histones are normally possibly charged protein because they have so many of arginine or lysine, those basic residues, amino acids. Okay, so why do they have two positively charged amino acid? Because think about it, your DNA is negatively charged because of a phosphate sugar. So phosphate P minus is negatively charged, so they can have ionic interaction. The DNA negatively charged molecules through the phosphate sugar backbone, you can make ionic bond with those arginine and lysine rich residues out of these histone proteins. Okay, so there are 8 histone proteins. We call them H2A, H2B, H3, H4, and then you double that. Okay, and then there's H1 kind of sitting outside. So H1 protein right there. It's kind of a concatenating, it's holding this together. So DNA goes around twice. So H1 protein and nucleosome. Okay, histone core. So that's the nucleosome structure. So the DNA not packaged into nucleosome, okay, engaged in gene expression. So DNA that's going around, normally that is not accessible for other proteins. Okay, if DNA to be engaged in gene expression or replication or recombination, okay, some part of DNA should be accessible. Okay, so it should be outside of the histone nucleosome structure. That's what it means. Okay, so they are not typically associated with the non-histone proteins, and they are involved in cellular processes. So there's accessibility issues. Maybe there may be the area where maybe replication origin is starting. So that's the unzipping area. That's where the gene promoter sequence is found. So transcription factor proteins can come and bind. Okay, so that's what it means. And nucleosomal DNA forms 30 nanometer fiber. Okay, so we're talking about that 30 nanometer fiber. Okay, so it's like those kind of structures. And 30 nanometer fibers are held together at the base by the nuclear scaffold proteins. Those are proteinous structure, scaffold protein, maybe something like that. Or is shown on the next figure. Yeah, there you go. There's a scaffold protein. Okay, nucleosome binding with the scaffold proteins, right? So those are non-histone proteins holding DNA looped together, and those are scaffolds. Okay. So it's a very complex structure. Okay, making DNA into very condensed form. That's what you need to understand. And then so that's the general introduction about the chromosomal structure found from the cell. Now let's talk about DNA replication. So DNA replication is a way to make more DNA. before a cell division will take place, right? So if you look at a cell cycle is what? If there's a single cell, it doesn't just stay there until the end of its life cycle. Instead, it may go through different phases, right? So there may be phage, okay? Seemingly, nothing happening, but there are a lot of things happening. And then mitotic phage, that's when a cell is going through a cell division. One cell becomes two. Okay, so there's a mitotic phage and phage. Okay, between the mitosis, that's why it's called interphage, between the mitosis. So anyway, so mitotic phage is taking a very small segment of the entire cell cycle. So normally cells stay in so-called phage. Okay, if you look at the phage, there are G1 cell growth phage and S phage, the synthesis phage. There are a lot of replication, duplication happening in the S phage, and then G2 phage. That's the gap 2 phage where a cell will prepare to divide, go into the mitotic phage. Okay, so the replication, DNA replication, where does it take place? It is taking place during the S phage of the phage in the cell cycle. Okay, understand that, because you have to have DNA duplicated first to be able to go through mitosis. Okay, and then if you look at the prokaryotic genome, okay, you will be able to find the place called origins of replication. Okay, origin of replication, that's where the replication will start. Another name for that is a replicon or the origin of replication. So they have a unique DNA sequence. Okay, normally that sequence is recognized by a special protein called replicator. Okay, so they may have different names, but let's call them just replicator. Okay, when the replicator comes in and bind, that's the beginning of the replication process. Okay, the protein recognize the replicator is called initiator. So initiator protein recognizing replicator, I'm sorry. Okay, the replicator is a DNA element, initiator protein element. Okay, and then the activity of initiator is regulated by the ATP binding and hydrolysis. Okay, that's what it says. So typically the replicator DNA element, replicon sequence, there are certain unique sequence bits found. Okay, especially if you look at E. coli, we call the replicator sequence as ORIC region, ORIC, and typically in E. coli ORIC, there are these repetitive elements, sequence elements. Example, especially within that 245 base pair long sequence, you will find about 5 repetitive sequences that are recognized and bound by a special initiator protein called DNA. DNA is the name of a protein which functions as an initiator. Okay, so DNA protein comes and recognizes these 9 more. 9 more means it's made up with 9 nucleotides. Okay, so each segment is made up with 9 nucleotides. Okay, so they are DNA protein binding sites. That's what it is. And there are also these 13 nucleotide long sequences found. Okay, actually this is a very AT rich sequence where the hydrogen bonds are broken up, so they become single strand DNA, kind of a double strand DNA is becoming single strand around that region. That's what it does, because they have a lot of A and T's in their sequences. Okay, so the first thing what happens is in E. coli, so DNA protein binds with the 9 more sequences right here. Okay, so there are multiple DNA proteins come and bind on those 9 more sequences. Okay, and so when there's ATP associated DNA, they interact with the DNA, the 9 more sequence. Once that's done, this DNA protein can recruit additional proteins like DNA B, which serve as a helicase enzyme function. Helicase does what? It's opening up the DNA, double strand into single strand form. So yes, the DNA A protein recruits DNA B, and then DNA C is functioning as a helicase enzyme loader protein. So you see these circular proteins, those are loader proteins. Okay, so that's what they do. So DNA C keeps DNA B in an inactive form. So they have different unique functions, as it looks like. So again, DNA B recruits, once that's done, DNA B, which is a helicase enzyme, that recruits another enzyme that's called RNA primase. Remember, DNA polymerase cannot start its synthesis unless there is a primer made out of RNA is made available, and that primer is made available by this RNA primase enzyme. That's RNA synthesizing enzyme. And then, so DNA B is recruiting that enzyme. That's what it says. Okay. So it's described here. There is that primase enzyme making that primer. So it's going this way. Then another one here. Primase enzyme is making a primer. Probably it's going to go 5' to 3' direction. Maybe it's this way. Okay. And then once that's done, the DNA polymerase 3 holo enzyme is brought to the origin. So we're talking about these guys. These are the holo enzyme, DNA polymerase 3 holo enzyme. Holo means a huge enzymatic complex. It has 3, small subunits, and each subunit can replicate DNA strand. Okay, so it's a trimer. Okay, so that's what happens. So let's see. So there's a leading strand sequence. So like here, so it's going to be leading strand replication here. There's a primer there at the end of the primer. So DNA polymerase grabs the tail of the primer and synthesize the leading strand. So it's going to continue as soon as this is opening up. It's going to continue that replication, right? And then there's a lagging strand synthesis, which is kind of a opposite orientation. So if this holo enzyme is going to be responsible for synthesizing both leading strand and lagging strand, there's a opposite orientation of this DNA templates. So the only way of replicating the same direction towards this way, this template DNA, the lagging strand template DNA, should be looped around like this, almost like a 180 degree. So then it can go same way from 5' to 3' direction, so towards this direction. We'll talk about that later. So there's actually looping going around on the lagging strand. So that's how this much of the Okazaki fragment is replicated from 5' to 3' direction. So DNA polymerase can only go from 5' to 3' direction, regardless of its leading strand or lagging strand. So here what's shown is there's only one DNA polymerase 3 holo enzyme replicating both leading and lagging strand. So for it to move towards the same direction, leading strand there's no problem, but the lagging strand should loop around 180 degrees. So that's how it can go towards the same direction. Okay. Same thing shown up here. Okay, so you can see DNA, protein, ATP, and the binding and recruiting DNA B, helicase enzyme, and the loader enzyme. So DNA C is a helicase. It says it's a loader, and DNA B is a helicase. They are recruited. So now unzipping just took place after DNA binding. Unzipping here. Double strand becomes single stranded, right? And then primase enzyme is recruited. Now they are making the primers, RNA primers. Once that's done, then holo enzyme, polymerase 3, is recruited. Now it grabs the tail of primer, 3' end, and continue its leading strand, continuous replication. This side, and then on the lagging strand, there's a looping that allows the same directional replication on the same holo enzyme. That's what is shown up there. Meanwhile, there's a sliding clamp that's engaged, so it'll allow the moving of the holo enzyme along the template DNA. We'll talk about that later on. Okay.